Okay, hi everyone. We're going to right now go over the second part of the cannabis lecture. Some of these slides we have already gone over in class, but a few we haven't. So I'm going to go over the slides that we went over in class one more time and then get to the ones that we haven't covered yet. Okay, so let's begin with this slide here. For this one, I would just like you to read over the information that's on this slide. So for instance, go over what the acute effects of marijuana are and what the chronic effects are. Some of this is going to be reviewed for you. So the important thing here is to see how the acute effect compares with the chronic effects. Something that you're going to notice as you look over this table are words such as possibly and likely. Right? So as you look at the effects, the chronic effects, and as you look at the recovery of function with abstinence, you see, possibly and likely stand out. So what I'm going to go over with you is why we see these words. What do they mean? So when you see words like possibly and likely, what it means is that previous evidence, previous research is mixed with respect to that particular function, right? Not all studies are in agreement. And the reason why you may have this variability across these different imaging MRI findings is that studies are going to differ based on some important factors. So what are these factors? So some of these factors could be, for instance, the amounts and pattern of cannabis usage. So participants are going to differ from one another. Right, so a particular sample, for instance, may differ from another sample based on who makes up that sample, right? And these participants are going to vary based on, their history of cannabis use and how they went about their cannabis usage. Was it, was it every day? Was it, weekly? Just every weekend? How much did they use every day or every week? When did they start their cannabis usage? At what age did they begin smoking, let's say, marijuana? How long have they been smoking for? Or have they been using cannabis for? Or how long did they have to not use cannabis before participating in a particular study? Different studies are going to have different criteria, right? So some may need you, for example, to not use cannabis at least 24 hours before participating. Some may have this length be longer. So there are going to be these differences across studies that could contribute the variability of findings. Now, despite a range of MRI findings that are not always consistent, several consistent findings have emerged. So what are some of these consistent findings? Okay, so one of the most consistent acute and chronic outcomes of cannabis use is memory deficits. So it's no surprise that the hippocampus, which is an area that's essential for memory functions, to be implicated. It also makes sense for the hippocampus to be involved given our previous discussion that CB1 receptors that are activated by THC in marijuana are highly expressed by this brain region. So a consistent finding is a negative association between hippocampal volume or size and cannabis use. In other words, long-term heavy cannabis use has been found to be linked to a loss of volume in the hippocampus. So with that said, given this structural change, it is no surprise that one of the chronic effects listed in the previous slide to be impaired verbal learning and memory. Cannabis users perform significantly worse than controls on neuropsychological tests of verbal learning and memory. Besides the hippocampus, other studies have shown structural abnormalities in the frontal regions of the brain, as well as the corpus callosum, the cerebellum, the amygdala, among other regions. Studies like this are showing us that cannabis use, right, long-term heavy cannabis use may result in long-term structural and functional changes. So it looks like there is some neurotoxic effect, as demonstrated by the loss of hippocampal volume in this particular image or example that I've provided in this slide. In this study, the reference for it is also provided. So if you ever want to see any of these studies that are showing structural and functional changes, feel free to let me know and I'll be happy to send you these studies. Okay, but with that in mind, it's, as we discussed before, it's always important to remember that the alterations in brain regions may be a result of a polydrug effect, right? So cannabis users may also be consuming other substances like alcohol and nicotine that previous studies may have not adequately controlled for. So it could be a combination effect that's resulting in these structural and functional changes. Or it could be also that some of these brain differences may have preceded rather than followed cannabis use. So it's important to keep these things in mind. However, across all of these different studies, we are seeing this strong association between cannabis use and, structural and functional changes. But, we always want to take it with a grain of salt because these other factors may also be explaining the variance observed. Okay, so data so far also suggests or also shows that exposure to cannabis during adolescence may be more harmful than during adulthood. So the earlier age of onset has been shown to be associated with greater cognitive impairment and more pronounced morphological alterations. And that's because normative neuromaturational processes become disrupted by cannabis use. So prolonged use during adolescence appears to interrupt how this very widespread endocannabinoid system is supposed to work. Marijuana or cannabis use, you can think of it as hijacking the brain's ability to fine-tune its synaptic connections as deemed appropriate by the nervous system, right? So because instead of our nervous system activating the CB1 receptors when appropriate, the CB1 receptors are out of context becoming activated by drug use. And all this is happening during a time when a lot of brain related growth, maturational processes is taking place and milestones are trying to be reached. So in a way, when marijuana is used very early, right? So during the time when we're going through puberty and adolescence, we find that the endocannabinoid system will no longer function in a way that it's supposed to function to support the normative maturational processes, because it is becoming active, it is participating out of context. So the nervous system will lose its ability to recruit it when appropriate to hit the necessary milestones. So let me give you a little background on the endocannabinoid system with respect to brain development. Okay, if you remember, CB1 receptors are widely distributed and expressed by many brain regions. Now, this distribution stays relatively constant across our development, but the density of CB1 receptors do change over time. The density and binding of CB1 receptors are highest during puberty onset and adolescence, and then the density of it decreases by adulthood. So there's a high demand for them during our early developmental stage. We see this shift in CB1 density going from higher to lower density, especially in areas of the prefrontal cortex. So the most dramatic and progressive decrease in CB1 expression across adolescence is seen in prefrontal cortical regions. And this is a decline that occurs in tandem with an increase in anandamide and a decrease in 2-AG within the prefrontal cortex. So the appropriate functioning of the endocannabinoid system appears to be very essential and important for the developing prefrontal cortex. Now, so it would make sense why adolescents and emerging adults may be at the greatest risk for the negative structural and functional effects of cannabis exposure, mainly because through THC, the brain's own fine-tuning ability at a very sensitive time period becomes disrupted. By constantly activating CB1 receptors and therefore desensitizing and downregulating them, the various neuronal populations then lose their ability to fine-tune and control synaptic transmission when appropriate. And in this way, neuromaturational processes may become compromised. Right? So this is why exposure during adolescence may be or is perhaps more harmful than during adulthood. Okay, I know you know this, but just a quick review. When I say fine-tuning and harmonizing and balancing synaptic transmission, that's because, based on our class discussion, CB1 receptors are involved in retrograde transmission. Right? So our endocannabinoid system is there to maintain balance because endocannabinoids are made on demand by the postsynaptic cell to activate CB1 receptors in the brain that will then work to prevent excessive neuronal activity. So our body's endogenous cannabinoids, or endocannabinoids like 2-AG, are the postsynaptic cell's signaling mechanism to disable the presynaptic cell. And in doing so, activity between cells can achieve some balance. So whenever the presynaptic cell is too active, the postsynaptic cell, through retrograde signaling, can silence that presynaptic activity so that you don't have excessive inhibition or excessive excitation. Right? Or when you find these CB1 receptors postsynaptically, again, their activation serves to reduce the depolarization and excitability of the postsynaptic cell. So they're there to create harmony and balance within the various neuronal or across the various neuronal populations. Okay, so lastly, we also see performance in certain cognitive tasks to be similar between cannabis users and non-users. However, in these cases, what we also find is that the level of activation in brain regions associated with the particular examined task, so let's say areas of the prefrontal cortex for working memory functioning, to actually be increased in cannabis users than non-users. So cannabis users are activating the particular brain regions associated with a particular task to a much greater degree than non-cannabis users in order to achieve the same performance. So this shows that greater neural effort is required on the part of the cannabis users to achieve accurate task performance or to achieve the same level of performance as non-cannabis users who are not activating their brain regions to that same level. So they can get the job done with less activation. So, and cannabis users additionally, when performance is the same between cannabis users and non-users, they also show increased activity in regions of the brain that are not typically associated with performance on a particular task, which again may indicate that their brain is switching strategies in order to compensate and to meet the cognitive demands of a particular task. Okay, so this slide here isn't showing you brain activation of cannabis versus non-cannabis users. Instead, it's showing you older versus younger adults performing a particular working memory task. But the reason I'm showing you this slide is so that I can demonstrate to you how the brain can change its level of activation, or it can go through recruiting different brain regions to get a particular task done as a means of compensation. So in this case, older adults are activating a particular brain region to a higher degree or recruiting additional degrees to compensate for normal age-related cognitive decline. In the case of cannabis users, they're trying to compensate for any structural or functional changes or damage as a result of cannabis use. So what you're looking at here is you see the brain activation of young adults at the very top, right? So to the top right-hand corner, you're seeing a unilateral brain activation of young adults when performing a particular working memory task, right? So on the bottom two, you see, so let's focus on the middle brain here, the second one. You're looking at older participants who are recruiting the same brain region when engaged in this particular working memory task. But as you see, they're engaging this brain region to a much greater degree, right? There's a much greater activation as demonstrated by a bigger red and orange area, right? So this red and orange blob is indicative of brain activation. And as you can see, it's bigger, which means that this particular region of the brain is getting activated to a greater degree to get the same task done. And then the third example are the case of particular older adults who can actually perform this particular working memory task at comparable levels to our younger adults by activating additional brain regions. So here they're not going through that same unilateral brain activation that younger adults go through. Instead, they're engaging in bilateral activation of their prefrontal cortices in order to compensate for their age-related cognitive declines so that they can maintain high levels of working memory performance. So the point of this slide is to show that the brain can compensate in different ways. It can either increase activity of a particular area or recruit additional areas to get the task done. So in long-term cannabis users, when performance doesn't suffer, so when they're performing at the same level as non-users, what is also seen is that their brain engages in more work. So it looks, in terms of more work, it could look like something like this second brain, the second one that's shown on the right-hand side. So there's greater activation that's done, or they can go ahead and switch strategies, just like the third example here on the right-hand side, by recruiting additional brain areas in order to compensate and in order to perform similarly to non-cannabis users. So to continue our discussion of long-term effects of cannabis use, an important topic to discuss would be cannabis dependence and what cannabis dependence is associated with in terms of more long-term type of structural changes in the brain. So most cannabis users do not go on to become dependent on cannabis and do not go on to seek treatments. Those that do become dependent, though, are going to experience withdrawal symptoms when they're not using cannabis, when they abstain from cannabis use. So withdrawal symptoms include irritability, increased anxiety, depressed mood, sleep disturbances, and decreased appetite, among other negative effects. Now, what is interesting here is that there's a physical alteration in our nervous system that has been identified to be related to withdrawal symptoms. And what I'm showing you is a figure from a particular PET imaging study that showed withdrawal symptoms to be negatively correlated with brain CB1 receptor availability. And in this study, it was in cannabis-dependent men. So let's go over this figure. The y-axis is showing you cannabis withdrawal assessment scale mean scores. So the y-axis is demonstrating the severity of withdrawal symptoms in participants. On the x-axis, you're looking at, you see how it says VT? VT is an index of receptor availability, and the name right before VT is a name of a particular or a selective CB1 receptor ligand. In other words, the x-axis is showing us a range of CB1 receptor availability. So what this figure is showing is that the higher the withdrawal symptoms in cannabis-dependent men after 2 days of drug abstinence. So they chose 2 days because this is the time of peak withdrawal symptoms, the lower the observed CB1 receptor availability. So results demonstrate that CB1 receptor downregulation is linked to a greater dependence and therefore withdrawal symptoms in chronic cannabis users. So, based on one's pattern of use, duration of use, the strength of cannabis, one can become dependent on cannabis as demonstrated by physiological changes in response to drug use. And whenever we change our nervous system, of course we will be experiencing withdrawal symptoms when not using the drug, because our body has adapted its functioning to the presence of the drug. And without the drug, we're going to be experiencing withdrawal symptoms. As a way, it's our body's way of indicating that the drug needs to be present in our body to achieve some sense of balance and homeostasis, and therefore the absence of pain and suffering. Okay, so we looked at structural differences. Let's now look at neurochemical differences, or the neurochemical changes that can happen as a result of long-term cannabis use. Because previous research has shown there to be mood and motivation related differences in cannabis users compared to non-users, and given the involvement of the dopaminergic system in our mood and motivation, it makes sense why research has really invested time and attention in examining dopaminergic function across users and non-users of cannabis. So a recent research actually, I think was from 2014, showed that dopamine synthesis capacity is negatively associated with higher levels of cannabis use. This was a super strong association. We're looking at an R of negative 0.77 and a P of less than 0.001. And this lower dopamine synthesis was related to an earlier onset of use. And the lower dopamine synthesis was also related to higher scores on a particular apathy scales. So lower dopamine synthesis was related to greater apathy, to greater lack of interest, to greater indifference. So to put it all together, long-term cannabis users not only demonstrated lower dopamine synthesis, which was related to greater apathy, but this association was found to be even stronger in those who had an earlier onset of cannabis use. Now, in support of the finding that chronic cannabis use is linked to reduced dopamine synthesis are other studies who have used PET imaging to examine striatal response to amphetamines. So they're studying the striatum because this is a brain region that receives a lot of dopaminergic input. For instance, we have been talking about the ventral striatum a lot, which includes the nucleus accumbens, right? Or the dorsal striatum is the region that receives dopaminergic input from the substantia nigra. That's a pathway that's implicated in Parkinson's disease, right? So, also, as you know, amphetamines and the amphetamine mechanism of action involves releasing dopamine from axon terminals. So this amphetamine test can show us the level of dopamine release. So the finding here is that individuals who meet the criteria for cannabis abuse or dependence exhibited decreased striatal dopamine response to the amphetamine test. So when we compared cannabis users versus non-users in this amphetamine test, cannabis users demonstrated significantly less dopamine release in response to the amphetamine test than non-cannabis users. So this dopamine deficit in the amphetamine study was not only found in cannabis users, but it was also linked with lower performance on tests of attention, working memory, among other cognitive processes examined. Okay, so alterations in the dopaminergic system have been identified in long-term cannabis users. Now, the point of this slide here is to demonstrate why, these changes in the dopaminergic system in response to chronic cannabis use make sense. Okay, so alterations in the dopaminergic system make sense when we think about what excessive and prolonged THC exposure can do to the cannabinoid receptors, right? They become desensitized and downregulated. With that in mind, over time, perhaps dopamine cells lose the ability to control various incoming inputs, whether they're excitatory or inhibitory. So what this figure is showing you is an example of a dopamine cell that is getting both GABAergic, which is inhibitory input, and glutamatergic, which is excitatory input. And when either of these inputs, right, whether it's the inhibitory or the excitatory input, becomes excessive, the dopamine cell, as you can see in this image, can on demand synthesize endocannabinoids. So in this example, it's showing you anandamide or 2-AG, which will, in the case of 2-AG, in a retrograde manner, can inhibit that incoming signal from that presynaptic cell in order to reduce the stimulation received by the dopaminergic cell. So let me give you a more specific example. So imagine a case where there's too much excitatory input from the glutamatergic neuron here. In this case, the dopamine cell is becoming excessively active in response to it. But the dopamine cell doesn't need to continue to receive this excitation when this excitation is excessive. It can exert control over this situation. It can control the incoming input by synthesizing and releasing 2-AG in order to inhibit glutamate release from this glutamatergic cell. So the CB1 receptors on the glutamatergic cell, when they activate in response to the 2-AG released by dopamine, will work to inhibit glutamate release from the glutamatergic cell. And in this way, the connection between glutamate and dopamine will become balanced. Now imagine if someone is heavily using cannabis for a very long time. By constantly activating the CB1 receptors, in this case imagine the glutamatergic CB1 receptors, these receptors will eventually desensitize and downregulate, right? So this can also, it doesn't have to be specific to glutamate releasing cells, right? We can have this downregulation and desensitization of CB1 receptors that are present on any neurotransmitter system. And in this way, the potential consequence is that dopamine can no longer regulate the activity of these incoming signals, whether it's from glutamate or any other neurotransmitter system, and the potential long-term consequence can be the disruption of the functioning of this system, right? Because the way that this system is connected to other neurotransmitter systems has become disrupted, and over time, perhaps the dopaminergic system can get impaired or can become impaired. So the next question to address is what is the relationship between cannabis use and developing a psychotic disorder or psychotic symptoms? So immediate psychotic-like responses to cannabis, if they do happen, are usually transient, short-lived in nature. So the question is, can heavy, long-term marijuana use result in or significantly increase the risk for developing a psychotic disorder, such as schizophrenia, later in life? So besides that acute psychotic reaction that's short-lived, are there long-term chronic effects of cannabis in the form of developing, let's say, schizophrenia? So this issue has been addressed by at least 13 longitudinal studies so far. right? So these 13 different studies have tested whether there is a relationship between early marijuana smoking and a later onset of psychosis. These results were conducted in a number of different locations worldwide, which included Germany, the Netherlands, the United Kingdom, Sweden, New Zealand, and the United States. Of these 13 studies published so far, 10 of them have found a significantly increased risk of developing a psychotic illness such as schizophrenia or severe psychotic symptoms, while a trend in the same direction was reported in the other 3. Okay, so several factors appear to influence this relationship, though it's not a black and white relationship. The factors that influence this association include 1. The amount of cannabis use, 2. Cannabis content, so THC to CBD ratio, 3. Age of initiation of use, and 4. The presence of any pre-existing vulnerability to psychosis. So let's go through them. Okay, so the first one is greater usage of cannabis has consistently been associated with increased risk for psychosis. It increases the likelihood. Very high cannabis users have what seems to be 4 times the likelihood of developing a psychotic disorder compared to non-users. Now, the second point, higher THC content along with lower CBD content also increases the risk for psychosis. So cannabis with high levels of THC but very low CBD has been associated with a much greater risk of experiencing a psychotic episode compared to using low potency cannabis. And by potency, we're talking about THC content or no cannabis at all, right? So here, when I say low THC content, of course, keep in mind THC to CBD ratio. Now, the relevance of CBD content stems from evidence that this cannabinoid, this phytocannabinoid, in contrast to THC, appears to have antipsychotic properties. So another thing related to this that's interesting or important to keep in mind is that, as discussed in class, in recent years, selective breeding and cultivation practices have resulted in increasing THC levels while keeping CBD levels very low. Usually you find it less than 0.4%. Now this is extremely worrisome because the presence of CBD with its antipsychotic properties dose-dependently counterbalances the psychotropic effects of THC. Okay. The third point, initiation of cannabis use in early stage of our lives, so in early adolescence, also appears to increase the risk of developing a psychotic disorder. This goes back to what we talked about previously in this lecture, right? The reason for this appears to be that the still developing adolescent brain is a lot more vulnerable to the disruptive effects of repeated phytocannabinoid exposure than the adult brain. And lastly, the fourth point is clinical researchers have identified people that they call quote-unquote ultra-high-risk individuals. These individuals are already in a either pre-psychotic state, which would therefore mean that they have a significant likelihood of transitioning to a psychotic episode as a result of frequent use, or if someone already shows psychotic symptoms, continuing use after the onset of the psychosis has also been shown to increase the rate of relapse into a psychotic episode compared to those who discontinued their use. So everything that I just talked about is pretty much being shown by this figure here. So if we go over the figure together, we can, for instance, see starting from the left-hand side, the higher someone's genetic vulnerability to schizophrenia, right? So for instance, if they have a family history of schizophrenia, combined with going to the next stage, combined with initiation of use early in the course of one's neural development, and then that combined with using very potent, right, high THC and very low CBD ratio, combined with using using cannabis frequently, all of that can go on to combine and disrupt neurodevelopment. That can then manifest itself by the expression of psychosis. So it's not a black and white relationship. Cannabis doesn't cause schizophrenia or psychosis. This relationship, this association, takes into account different factors. And then when everything aligns in a particular way, then the likelihood increases. Okay. Now, when we talk about potency, on that note, you can imagine that synthetic cannabinoids like K2 or spice, they pose an even greater danger because of their much greater potency and ability, therefore, to precipitate into acute psychotic reactions. Okay, so now just to repeat some of these main points, as already mentioned, but maybe some of these points are perhaps new. Few researchers in the field that are examining the relationship between cannabis and psychosis, few of them believe that cannabis by itself causes schizophrenia or any psychotic disorder. Again, investigators hypothesize that cannabis use, particularly when started during early adolescence, then taken regularly in high potency forms, goes on to interact with these other factors to enhance the risk of development of later psychosis. But continued research in this area is of course needed. Why? Because the mechanism underlying the link between adolescent cannabis use and later risk for developing psychosis, right, the mechanisms are not yet understood. So with that in mind, whether there's even a causal connection, right, and to what extent, still needs to be determined, right? It's not determined yet. Okay, but let's say if there is a causal contribution, we can speculate that there's potential involvement of particular neurochemical systems that are known to be affected by cannabinoids that are also involved in the cause and the symptomatology of schizophrenia, two of which would be dopaminergic and glutamatergic systems. But anyhow, more research is absolutely needed in this area. Okay, so the next slide here, this is an FYI slide. Go ahead and read what's in the in the blue boxes and the red boxes, and you can see that two potential mechanisms have been proposed by that explain how prolonged cannabis exposure during adolescence might disrupt the function of the endocannabinoid system and therefore alter brain development. So the red pathway is showing us or talking about altering synaptic pruning, and the blue pathway is showing us alteration in the development of white matter. So this is FYI, please go over it if you're interested. Okay, so what are some of the other effects of cannabis use? So cannabis use can affect the reproductive system. We find that THC results in menstrual cycle irregularities in females, or at least in some females, reduced circulating testosterone levels and deficits in sperm concentration and motility in males. And related to the reproductive system is, of course, the issue of pregnancy. Specifically, the question is, does maternal cannabis use influence the developing child prenatally? So this is especially important to go over because given the progress in the legalization of marijuana, it's expected that there will be an increase in users due to greater access to the drug and, of course, the lessening of penalty for use and possession. Okay, so among those affected will be pregnant women. We find that marijuana seems to be the most frequently used illicit drug by pregnant women, and apparently it is also being advertised as a remedy for pregnancy-related nausea. So for us, the important question is, of course, does cannabis use during pregnancy have any risks to the growing child? And if so, what are these risks? So before we go over the results, remember that THC is lipophilic. Right, so with that in mind, you can imagine that it readily passes through the placenta into fetal circulation. You also know that the CB1 receptor is the most abundant metabotropic receptor in the mammalian brain. Okay, so of course, with that in mind, it's no surprise that the fetal brain expresses cannabinoid receptors. right, and a developing endocannabinoid system that could become disrupted by repeated exposure to phytocannabinoids such as THC. There are actually recent meta-analyses and reviews on maternal and neonatal outcomes that have found a significant association between maternal cannabis use and an increase in negative outcomes such as low birth weight of infants, smaller head circumference at birth, which indicates reduced brain size and predicts lower IQ later in life, and greater likelihood that the newborn will require placement in neonatal intensive care unit, among other negative effects. So these findings remain even after the data is controlled for maternal tobacco use, which itself is a major cause of fetal growth restriction. So in this case, growth restriction during fetal development is thought to be related in part to the activation of placental CB1 receptors, which result in decrease in nutrient and oxygen availability to the fetus. Now, longitudinal studies have also investigated the possible impact of prenatal cannabis exposure on behavior later in infancy and childhood. So findings depend on various factors. So depending on the age of the child, when assessment is conducted, depending on the amount of maternal cannabis use, depending on when mom smoked when pregnant, so during which trimester of pregnancy was the developing child exposed to cannabis, right? So depending on these various factors, we find various types of deficits in a number of important domains. So these domains are deficits in visual perceptual function, deficits in language skills, deficits in attention, in memory, and educational achievement. So now, of course, as always, keep in mind that everything we've been talking about and all these different effects that are displayed for you on this figure may not just be strictly attributed to the effects of cannabis use during pregnancy. Why? Because pregnant women who persist in using cannabis may also differ from non-users in other important ways, such as their level of stress exposure, their nutrition during pregnancy, factors related to socioeconomic status, or any other factors. So, pregnant mothers that use cannabis, again, may differ in many other important ways than pregnant mothers who don't use, and those other factors may also contribute to some of these effects that are observed. Okay, so in this lecture, we really focused on some of the negative effects of cannabis, especially in long-term heavy cannabis users, and especially in those that use very high potency strains. So with that said, of course, as you know, as our group presentation went over, as your book discusses, cannabis as a plant, we can't see it in a black and white manner. It's not all good, nor is it all bad. It really depends on that THC-CBD ratio, and it really depends on the pattern of one's use, right? So with that said, while there's one school of thought that there's certain individuals that really glorify weed, right? This miracle drug, this therapeutic, natural plant that's good for us, right? So there, you probably know, or you may be one of those individuals that may have held this idea. But based on the recent research that we have accumulated, we can't really deny some of these findings. We can't deny that we can't deny that dependence does not form, right? People, depending on their use, can become dependent on marijuana. We looked at how these CB1 receptors change, and we also looked at how this change in CB1 receptors can explain or is associated with the withdrawal symptoms. Or, we can't say that all strains are good, right? So the THC to CBD ratio does matter. There is danger in using synthetic cannabinoids like K2 or spice, right? Or we can't say that pregnant women can just smoke marijuana and have no consequence for the child, right? That's not supported by evidence gathered so far. So I'm not personally here to tell you that you should or shouldn't smoke. I'm not also here to say whether marijuana should or should not become legalized. But what I'm here to say is that in order to make decisions that benefit us, we need to be well informed so that we can make decisions accordingly, right? Maybe you're someone who does want to smoke marijuana, and in that case, perhaps you should look into and care about the THC and CBD content, right? Or perhaps it would be important for you to monitor your pattern of use so that you don't induce long-term negative structural and neurochemical changes in your brain. Or perhaps later on, if you do become a parent, it'll be important for you to be able to explain to your child, speak to your children about the consequences of their use, given that they're in a very developmentally sensitive time period with respect to their neurodevelopment. So now, we really focused on THC, especially because with the legalization of marijuana, a lot of people are going to be wanting to access cannabis mainly because of the THC content, so that they can experience getting high. Now, CBD and its therapeutic potential, right? So high CBD, very low or no THC is a whole nother story. Some of the slides that I've posted for you, at least I think a few of them focus on the effect of CBD on controlling seizure-related disorders in certain individuals. So feel free to take a look at that, read about it in your book. I will try to find some good reviews that go over CBD, its pharmacology, and the various therapeutic potentials it has, so that you could read it just FYI. But I'm going to end lecture here. I think, or I hope that we had, a full, meaningful discussion of THC and our cannabinoid system. And if you have any other questions, just let me know. Okay, see you guys soon.